So the coronavirus, how do they do the test? How do they screen for the presence or absence of coronavirus? The coronavirus test is all about the polymerase chain reaction, PCR. Let's take a quick look at this. We have a molecular biology group, and they want to study a promoter in a piece of DNA. The DNA looks like this. They're interested in the DNA from this spot to this spot. So they're interested in all of this. They need a primer on both sides of the DNA they're looking for. So the forward primer is this one right here. It's 20 bases long. It matches the 20 bases that we're looking at. We know the sequence of the DNA. This is an important foundation of this. We know what we're looking for. The forward primer is right here. And then we need a reverse primer. The reverse primer is right here. The reverse primer is the rails of the DNA are opposite each other, so we need the reverse primer is opposite. It's what they call the reverse complement. It's as if the two primers face each other. And then when you run the thermocycler, the polymerase chain reaction makes a copy. The next cycle makes another copy. The next cycle makes another copy. So the amount of the DNA they're looking for doubles with every repetition. It doesn't take very long to get up to millions and billions of copies. This is the traditional use for the polymerase chain reaction. So what does this have to do with the coronavirus? We're taking the test for the polymerase chain reaction. We're taking the polymerase chain reaction and we're using it to look for the DNA. The coronavirus pandemic came out of China. In a very short period of time, the Chinese scientists came up with a sequence for coronavirus. So we know what the DNA, it's very much like this. We know what the DNA is. So the idea is to make primers that are specific for this DNA and then run an experiment on the specimen from the patient and see if it works. Let's use an example. Teresa has been in Italy. She gets back, she gets off the airplane and she doesn't feel very well. She's concerned about the well-being of her children. She wonders if she has coronavirus. She goes to a testing facility, gets in line in her car, she gets to the front of the line, rolls down the window of her car. The technician hands her a little swab. She swabs out her mouth according to the protocol. The technician puts the swab back in its little house. The technician gives Teresa like a little receipt sort of thing, and Teresa drives off. The technician runs a test on the swab. She does an experiment on it. It's a polymerase chain reaction experiment on it. She does the experiment, and the experiment doesn't work. So that means that there isn't any coronavirus in it. The next sample comes in, comes back negative. The lab does 100 samples, and the experiment never works. Finally, the experiment works, and they go, oh yeah, right, this experiment worked. If the experiment works, it means that the coronavirus is present. They have primers that are specific for coronavirus DNA. We know what the DNA is of the coronavirus. We apply those primers to the specimen. If they work, then the coronavirus is present. If they don't work, then the coronavirus is not present. The analysis for the coronavirus is all about the primers. So what are you going to use as a target in the 30,000 bases? They like to use an mRNA target, a messenger RNA. And you think about it, it makes sense. Out of those 30,000 bases, which ones are important? Well, the ones that are important to the virus is the messenger RNA. And recall that the messenger RNA is a copy of the DNA, the original. The transcription of the DNA into RNA produces RNA, and the RNA is the same thing. But how are we going to put this into the polymerase chain reaction? Well, there's an enzyme called reverse transcriptase. The reverse transcriptase turns the messenger RNA into double-stranded DNA, and then it becomes regular polymerase chain reaction. So this is where the language RT-PCR comes in, reverse transcriptase polymerase chain reaction. I'm a pretty lazy guy, but I do research sometimes. So reverse transcriptase, what's that? I go to a textbook from 2001, Molecular Biology by Paul Weaver. It's a textbook that I used a long time ago. I look up reverse transcriptase. They got a long chapter in it. It was old stuff in 2001. The polymerase chain reaction has been around since the 1980s. This is not new technology. This is not something that's uncommon. The polymerase chain reaction, I'd be willing to bet you that there are very few molecular biology laboratories in America that don't do polymerase chain reaction. And then the reverse transcriptase, this is a very common technique for people that study mRNAs, which is a lot of people. The technology and the platform for doing the screen for the coronavirus, I would be willing to bet you that there are dozens of them in America. This is not a simple test. This is not straightforward. I'm not pretending that, but we have the facilities, we have the infrastructure to do this. The infrastructure consists of the equipment and the people that know how to do it. The screen for coronaviruses is all about the primers.
I think, like most of us, I want to understand things and be able to kind of wrap my head around things. So the primers, the primer goes out 20 bases long and hybridizes with a match it finds in the DNA. I mean, why would the primer do that? I mean, it didn't. The key to the understanding is this, for me. The The reaction vial for the polymerase chain reaction is a soup of primers and trinucleotides. It's primers, 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 trinucleotides, and then there's a little tiny bit of target DNA in it. So that the DNA, if there's a match between the primers and the DNA, the primers are going to find it because there's so many primers. And then the trinucleotides, the whole thing is driven by the trinucleotides, which is the adenine triphosphate, guanine triphosphate, cytosine triphosphate, thymine triphosphate. That's what drives the whole thing. It's like in the big picture, the target DNA is not really a big part of it. If there's a match between the DNA and the primers, there's so many primers, they're going to find their targets.